When it comes to eukaryotic gene regulation, we have a lot more places that this can occur. In prokaryotes, we saw it was mainly at the transcriptional level. We're going to talk about several different levels in eukaryotes, and we're going to start with what we call epigenetic gene regulation. So we're going to talk about chromatin remodeling. That's basically what the chromatin state is. Can you actually get in to do transcription? We'll talk about how this is controlled by histones, and then we'll talk about a process called DNA methylation that can lead to what we call epigenetic gene changes. So epigenetics is a change to the phenotype of an organism without a change to the genotype, and that's a little bit backwards to what Mendelian genetics classically taught us. But if we think about this, right, gene regulation is epigenetics, right? We have seen examples of this occurring, and I will show you a few simple examples that can occur. Let's look at where we're at here, right? We're at the top. We're looking at regulation of transcription due to chromatin state and basically how the DNA is structured here. We will, in future sections, look at the downstream bits of this. So let's begin with the chromatin. So remember, our DNA is wound around proteins that are called histones. And you can see this on electron micrograph. Here's a DNA strand, and all these little beads that are on the string there are the histone proteins. A group of histones and DNA forms what we call a nucleosome. The interesting thing is the DNA can slide around these histones. The DNA can get wound up or it can get unwound on these histones. So let's look at that, right? We said euchromatin is the state where it's unwound versus heterochromatin is where the DNA is tightly wound. What do you think this winding would do to transcription? Go ahead and pause the video, think about that. All right, so let's look at heterochromatin first. Now remember, transcription, we need RNA polymerase to get in there to actually transcribe the genes. If you have DNA wound around these giant histone proteins, RNA polymerase is not going to be able to get in there. Whereas in euchromatin, RNA polymerase can get into these open, unwound areas, and transcription could theoretically occur. It's not guaranteed to, but it can occur. Whereas in heterochromatin, it cannot occur. So this state, heterochromatin and euchromatin, can actually change, right? We've seen it where it changed when we had cell division, those X-shaped chromosomes. That's all heterochromatin. Obviously, that has to unwind. But one interesting area that we're learning about is there are enzymes that can change the chromatin state. And what causes that to happen and where it happens is still something that we're learning about. But let's look at a couple of ways that this can occur, not necessarily when they occur, but how it can occur. All right, so let's look at heterochromatin. Well, heterochromatin, we said, can't be transcribed because RNA polymerase and transcription factors can't fit in there. Well, there's a couple of ways that this can occur. One of them is by methylation of the histones. So a methyl is a CH4 group, and it can come in and get added to the histone proteins. When histones get methylated, it causes them to wind up and basically become heterochromatin, making the DNA inaccessible. Gene inactivation occurs. The opposite can happen when we put acetyl marks on there. These acetyls cause the histones basically to spread apart from one another, opening up the DNA, allowing access to it, causing euchromatin to happen. You could have transcription occur. These methyls and acetyls can be added and removed, and there's complicated regulation to this. Again, we're still learning about why and when this happens, but let's look at an example we've seen previously. So we talked about the phenomenon of X inactivation, which happens in biological females where there are two X chromosomes. So to keep gene dosage proper, one of the X chromosomes gets inactivated. It becomes heterochromatin. Well, how does that happen? Through chromatin histone methylation, right? That causes it to become heterochromatin. So we said if there was a orange fur color gene on the active X and the black fur color was on the inactive, right, it would show up in certain cells as orange fur color. But this process is random, so in other cells, the orange might be inactivated and the black might be active, causing black fur patches. So this is an example of where heterochromatin is affecting phenotype. There's no change in the genotype. They're still heterozygous here for the two different alleles, but their phenotype is different based on epigenetic changes. Okay, so that's not too mind-blowing, right? But there is a field that is still in its infancy and still very controversial called transgenerational epigenetics. We still don't know a lot about this. The theory is that, in theory, let's say there's a gene for pigmentation. It could be shut off by changes in the chromatin, right? So the maternal lineage is brown fur, and we could acquire a phenotype that shuts this down, right? Epigenetic changes could shut this down. In theory, this could be passed on or imprinted. So the genotype is still the same, but the phenotype is different, this shut off gene phenotype. It's kind of like the tortoiseshell cat, right? If that got passed on reliably. How this happens and what affects this is still in its infancy. So we're not quite at the Assassin's Creed level of epigenetic memory in your genome, right? You can't go in the Animus and relive your ancestors' memories. But this potentially could be very, very interesting to understand, right? Changes in phenotype without changing the DNA sequence, right? You're just changing whether it gets expressed or not. So those are changes through histones. We also have changes in the DNA itself. Now, DNA methylation doesn't change the bases, right? Here we have some G's and C's, but we can add marks to them called methyls that can change whether a promoter is active or inactive. When there's a lot of methyls on G's and C's in a promoter, we tend to find that the gene is not expressed, whereas when those methyls are removed, the gene is expressed. Again, what controls this and how we can change that, we still don't have a lot of good tools to do this, but we are starting to learn. And this field is still in its infancy, so adding methyls can repress gene expression, whereas removing the methyls can activate gene expression. All right, so we had two epigenetic ways genes expression can be silenced. What were those two? Go ahead and pause the video and think about that. Okay, luckily they're the same mark. We said histone methylation would cause heterochromatin, which would silence gene expression. And we also said methylation of the DNA in the promoter would silence gene expression as well. Very interesting field that we're still learning a lot about. All right, so epigenetics, as we said, involves a change in phenotype without actually changing the DNA sequence in the genotype. This is due to changes in gene expression, right? So whether a gene is on or off affects the phenotype. Chromatin is one way this can happen. So DNA is wound around our histone proteins to form those nucleosomes. We said heterochromatin is tightly wound. Genes are not expressed in heterochromatin, whereas Euchromatin is more loosely wound and genes can be expressed there. There were kind of two ways we could go about this. Histone modification can affect this. So you can methylate a histone to create heterochromatin, or you can acetylate histones by adding acetyl marks to cause euchromatin to happen. We also said that DNA itself could get methylated. And if we see DNA methylation in promoters, that tends to prevent transcription from occurring. All right, so that's our epigenetics. This is still a new field and we don't fully understand it, so it's going to change and evolve as we continue to make new breakthroughs.